Welcome, everybody. I think this is the first Jetson call of 2025. It seems a bit late to say Happy New Year, but Happy New Year to you all. Um, I'm delighted we have two excellent speakers, as ever. And without further ado, I'm going to hand over to Randy Bundy, who's going to talk to us about iron availability and heterotrophic bacteria in the twilight zone. So over to you, Randy, with thanks.
Great. Thank you. Thanks for having me. I'm excited to talk about this work today that we've been doing on looking at effects of iron on heterotrophic bacteria in the mesopelagic. And first, I want to acknowledge all of these wonderful co-authors. And importantly, these are the kind of heroes of this work. And most of what I'm going to be showing was done by these lovely folks, especially Travis Mellett, who was a postdoc in my lab and is now a research scientist in the lab. So most of what I'm showing will be from his work today. Oops. Um, so as many of you know, iron is required for several key metabolic processes in phytoplankton, and we've been thinking about this for a long time in the ocean, particularly in the trace metal community, which is kind of my main background and focus in oceanography. And so we know that many different metals are cofactors and important enzymes, and iron is one of these, and it's used a lot for example, nitrogen uptake, but also heavily in photosynthesis as well. But iron is also a major requirement for heterotrophic bacteria as well. And we've been a little less focused on this, I would say, overall in the field of trace metal biogeochemistry, at least. But for heterotrophic bacteria, they tend to use iron mostly in respiration pathways. And from the estimates we have, we think that about upwards of 90% of their intracellular iron is mostly used in these kind of like functional proteins in the citric acid cycle. So they also need iron along with phytoplankton. And I think one of the reasons that maybe we haven't focused on this as much in our field is that we think that based on some of the estimates that we have, this is kind of a table summarizing average kind of iron quotas or requirements for different phytoplankton, bacteria, and also zooplankton. And we tend to express these kind of like a Redfield ratio. So here is a table of iron to carbon ratios in micromoles per mole that we think of are kind of representative on average of different groups, including heterotrophic bacteria. And one of the previous estimates that we've had is this 7.5 number, and this is from Phil Tortel in the 90s. And relative to other groups, they essentially have relatively high iron requirements, but maybe less so than the average cyanobacteria, for example, but more than maybe the average diatom or other eukaryotic phytoplankton. So they kind of, from this initial estimate, we thought they maybe have kind of a mid-range iron quota. And I'm going to come back to this 7.5 iron to carbon ratio. So try to think about that later in the talk. But this is kind of what we've understood previously about how much iron heterotrophic bacteria might need. And so what we're kind of interested lately in my group is thinking not just about kind of the amount of photosynthesis that can occur in the ocean based on kind of, iron maybe limiting growth of phytoplankton since we know that they need a lot of iron, but we're kind of interested in what's going on with the heterotrophic bacteria and more of this kind of respiration, remineralization side of the biological pump. And for the purposes of this talk, we'll think about just that part of it that's going on in kind of the dark ocean, or at least the upper mesopelagic for the work that we've been doing so far. And so we want to know what's the role of iron in that part of the biological pump. And one of the ways that we got interested in this in my lab is I am coming from a background of studying trace metal biogeochemistry and also organic molecules that complex to trace metals. And one of these compounds or classes of compounds are called siderophores. And these are some example marine siderophores here. And these are organic ligands that are really, really strong complexers of iron. And so they bind to iron super tightly, and they're produced purposely by bacteria or fungi for iron acquisition. So they're interesting because this is a way that we can kind of link specific kind of biomarkers to certain microbes because we can look at the genes used by different bacteria, for example, to produce these different compounds. And in general, what we've seen in other environments is that they're typically found where dissolved iron or iron in general is kind of scarce or in some really inaccessible particulate form. So, for example, we mostly knew about siderophores first from kind of pathogenic systems and also in soil bacteria. And they're produced in soil environments not because there isn't a lot of iron around them, but it's in this kind of particulate form that's not easily taken up and accessible. And these siderophores can kind of solubilize some of that iron, make it into a form that's more easily taken up into the cell. And so, in general, in the ocean, we've been thinking about these as kind of biomarkers for where dissolved iron might be sort of not very bioavailable or where it's kind of scarce, and that organisms would devote energy and resources to producing these specific compounds. So we were interested in these compounds and we've been trying to measure them in the ocean for a while, and it's pretty tricky because they're low abundance and we're in this, very salty matrix, et cetera. But when I was a postdoc with Dan Rapetta, I was doing some work at Station Aloha, and this was when we were kind of first developing a lot of these methods still. And I looked at a profile at Station Aloha just from surface to 450 meters, which I'm showing here. And there's different amounts of data on this plot, but the main thing that I kind of want to point out here is in this middle panel, we have kind of a concentration of organic ligands that could bind iron that we could extract with our solid phase extraction technique. And so those kind of increase with depth following a general iron profile. But of those ligands that we could extract, we can measure some specific classes of siderophores, which is on this kind of furthest right panel. So we were measuring two classes at this time, ferrioxamines and amphibactins. And we were pretty surprised that actually the highest concentrations that we saw of these compounds were in kind of the upper mesopelagic zone, up to about 11 picomolar. So these are small concentrations, but this profile was really surprising to us because we didn't think at all that the siderophores would be higher, deeper in the water column as dissolved iron concentrations are kind of generally slowly increasing. Although this is in a zone where dissolved iron is typically still very low and kind of the ferrocline is a bit deeper here. But this was super surprising to us because there's a lot lower bacterial biomass here. We thought most of the action would be in the surface waters. So this was kind of interesting. And when we measured this first profile, we then did a few kind of follow-up experiments on later hot cruises at Station Aloha, where we saw that siderophores were indeed kind of produced if we took sinking particles and kind of added it to free living bacteria living at these depths. And so we at least started to think about, okay, maybe there's something associated with kind of processing the sinking organic matter that would lead mesopelagic bacteria to produce these compounds. And so that's what kind of got me interested in pursuing this further. And so I went on later to do some different uptake experiments also at Station Aloha with iron bound to different siderophores. And so what I'm showing here is on the bottom, we have an inorganic iron addition that was added to some incubations in the form of radioactive iron 55. So it's just spiked in kind of iron chloride. And then we have three different siderophore bound iron treatments, which were also kind of radioactive iron bound to different siderophore compounds. And I did experiments in the surface at 15 meters in the light red, and then in the mesopelagic in the dark red at 300 meters. And the thing that was very surprising here as well is that we saw really fast uptake leading to these kind of shorter turnover times of iron in the mesopelagic relative to the surface. And it was also very fast for all of the siderophore bound iron treatments. And so this also is very interesting, again, just because there's a lot lower bacterial biomass at these depths. So the really quick uptake was pretty surprising. And I don't have time to show it here, but we also did some more siderophore profiles at Station Aloha along with this work. And we seasonally saw kind of these high siderophore concentrations in the mesopelagic again, like I had seen in that kind of first profile. So we wanted to also kind of, since we saw these fast uptake rates, we wanted to try to estimate, okay, well, what are kind of the iron uptake quotas that we're seeing in the mesopelagic at these depths, since there's so, many fewer bacteria. And that's what I'm showing here on the left. So this is now iron to carbon kind of uptake ratios that I was calculating from those radioactive iron uptake experiments, and then the different iron additions or substrates that I was using are on the x-axis here. And when I showed you in the beginning our kind of one published estimate of iron to carbon quotas for marine heterotrophic bacteria, it was about 7.5 micromoles per mole. And when we calculated our ratios in the surface waters, we got a range of 6 to 67. So it kind of encompassed this estimate, but we also got a lot higher values on the high end here. But then our values in the mesopelagic were just so high. We had iron to carbon ratios of 88 to 710 on average micromoles per mole across these treatments. And so this was very, very high, and I was actually like pretty scared by these numbers, and I wasn't sure if they were right or what was going on, but it was kind of intriguing that perhaps mesopelagic bacteria have really, really high iron quotas for some reason or another. And so all of this together kind of led me to think about, well, what is going on kind of with iron cycling in the mesopelagic? You know, we had certain lines of evidence together with the siderophore data and the iron uptake data that there's something going on with bacteria here where they need a lot of iron or they're taking up a lot of iron or there's really rapid iron cycling. And so we had these hypotheses that we wanted to explore, which were that potentially mesopelagic bacteria need to use more iron to process less labile organic carbon in the mesopelagic that they might be exposed to as opposed to surface waters. And another hypothesis we had was that mesopelagic bacteria might just be kind of storing iron intracellularly. Maybe it's kind of luxury uptake until they receive some kind of, pulse or input of organic carbon that they then can kind of use that iron to process that organic carbon. And we were interested in exploring this because we think that the iron supply then could be impacting bacteria growth and maybe their growth efficiency. And this we think is related to kind of the efficiency of carbon export in the mesopelagic. So that's why we wanted to explore this. So we went about exploring this in the field on some cruises that were called the Gradients cruises, and these are funded by the Simons Foundation. And there's 5 cruises overall in the project, but we did these experiments just on these last 2. So they were called Gradients 4 and Gradients 5, and they left from San Diego, went down just south of the equator, and then back into Honolulu. So we were crossing some nice gradients and just chlorophyll and inorganic nutrient supply and also iron supply. So on these cruises, we did several different experiments, and we did them in 2 locations on each cruise, in the subtropical gyre and then one in the equator. And in these experiments, we chose several depths, either 2 or 3 depths, depending on the cruise and the experiment. So I'm just showing you kind of one example experimental setup here for the first expedition, Gradients 4. And so we did incubations at 3 different depths within kind of the upper mesopelagic. We separated out some larger grazers that could be there with a 7-micron mesh, and then we incubated our trace metal clean seawater in the dark for about 48 hours. And our treatments were an unamended control, and then we had 1 plus iron treatment where we added 1 nanomolar iron 57, iron chloride. And then we had a plus DOC treatment, which was 60 micromolar glucose. And then we had an iron and DOC treatment, which was the 2 of them together. And we sampled for a suite of things, including dissolved and particulate trace metals to look at iron quotas, flow cytometry, DOC, nutrients, and siderophores. And so we called these our kind of high organic carbon treatments because we added 60 micromolar labile DOC. And then on the second expedition, we kind of adjusted our experimental protocol. We tended to do only 2 depths at our different locations on this cruise because we had more treatments. And we had the same treatments, kind of the iron, the glucose treatment, iron and glucose, but then we added 2 additional treatments in glucosamine and then an iron and glucosamine. And glucosamine is just kind of a slightly less labile form of glucose. And we also added these in lower concentrations, so 10 micromolar of the labile DOC in the form of glucose and 10 micromolar semi-labile. And so we call these the low organic carbon experiments. So I'm going to just show you some example data from these experiments, not overwhelm you with all of it, and then I'll kind of show all of the data together. So the first one that I'm going to show is just an example from the first expedition, this high organic carbon treatment from gradients 4. And on the left is just a profile from the subtropical gyre where I'm showing this example experimental data. And where these dashed lines are the depths where we did the experiments at the 3 different depths. And this is just to kind of show that this is where heterotrophs trophic biomass was present. We still had in our kind of 2 shallow steps some amount of oxygen, and then we get down to lower oxygen in the 450-meter depth, for example. And then what I'm showing in the plots is the treatments are on the x-axis, and then we have on the left column the kind of total carbon biomass from heterotrophic bacteria, and on the right the net growth rates. And basically what we saw in all of these steps was that there was higher bacteria biomass and growth rates in all of the plus iron and carbon treatments together. But a thing that was kind of interesting was that at some of the depths, carbon alone did not yield more growth. So for example, you could look at 450 meters, there's barely an increase kind of in the glucose treatment. And also if we look kind of more at 250 meters, the net growth rate in just the glucose alone, and then iron and glucose alone are very similar. So we saw kind of depth variations in whether carbon alone stimulated a response and whether you had to have iron and carbon together to get a response. And that was kind of a common theme amongst these experiments. I'm going to show you one example also from the second expedition where we did the lower organic carbon treatments. This time, this is an example from the equator at our 2 depths, and kind of the oceanographic context is shown here on the left. And so this one, we had more treatments. This is the one where we have glucose, iron and glucose, but then also glucosamine, which we've labeled with this little M at the end of it in the treatments on the x-axis. And we saw similar things here where we also saw higher bacterial biomass and growth rates in kind of the plus glucose and iron and glucose treatments. And more growth was observed when iron was added along with glucosamine. So we saw the most growth when we added glucose, essentially. There was a little bit of a response with the less labile organic carbon substrate. And in some cases during this expedition, when we added iron along with the less labile organic substrate, along with glucosamine, we saw slightly higher net growth rates, but not as high as when we added the more labile substrate. And again, on this cruise, we also saw some small variations in responses at the different depth horizons, whether kind of you would interpret it as iron maybe limiting their growth or just carbon, or iron and carbon co-limiting. So there was kind of interesting depth variations there. So I know I just showed you a few examples. I'm going to now show you kind of all the data together from all experiments so we can start looking at some patterns. So these are going to be more in box plots. And so what I'm showing here now is on the left column are the high organic carbon treatments. On the right are the low organic carbon treatments. And then the top row is just the biomass fold change in bacteria that we observed. And the bottom is the net growth rates that we observed. So this is just showing kind of like a summary of all the responses that we saw across depths and treatments from both expeditions. And essentially, we see similar things that I've already pointed out. Most of the largest kind of biomass fold changes were when we added iron and carbon together, although that was less true when there was these low organic carbon treatments. There was less of an effect with iron. And then the highest net growth rates also were when iron and carbon were added together when carbon was plentiful. So iron mostly had an effect when organic carbon was replete and sufficient, although again, there were some differences within certain narrow depth horizons of whether or not there was an iron effect or not, or if there was a co-limiting effect. Okay, the other thing that we saw that was really interesting in this experiments was we were looking at siderophore production in different treatments. And what I'm showing here is just kind of 2 depth examples. So the top row is from the subtropical gyre at 250 meters. The bottom is the equator at a similar depth, 300 meters. And then the colors are the different treatments. And wherever you see a peak in these chromatograms, that's representative of a siderophore that we could identify with mass spectrometry. And so you can kind of see in like the lighter green blue colors, that's where we see the most peaks and the biggest peaks. And those are always when we added glucose. And so if we pare this down to just kind of the amount or concentration of siderophores that were produced on a per cell basis, that's what the bar charts are over here. We saw kind of this like outsized siderophore production when we had added a bunch of glucose. So when we added a lot of carbon, this is in the high carbon treatments. And we saw a lot of these kind of types of siderophores that we've seen in the marine environment before. And now I'm going to put that kind of siderophore production data here on the bottom. And then on top of it, we have bacteria growth efficiency. And what was interesting is that we saw higher bacterial growth efficiency when iron was added in our kind of high carbon or replete carbon treatments. We didn't really see that effect in low carbon. And we saw lower bacteria growth efficiency when we saw really high siderophore production. So in these kind of just carbon treatments where we saw a lot of siderophore production per cell, that's where we saw lower bacterial growth efficiency. Okay, I'm going to try to put some of these things together now. And so the last bit that I think helps us understand what happened is looking at what happened with the iron quotas in these experiments as well. And here I'm collapsing all of the data from all the treatments and cruises into one plot, but we're looking at iron to carbon that we observed in bacterial biomass versus the total amount of bacterial biomass that was present in the experiment. The colors are based on the treatments. And the first takeaway was that we saw a really huge range in iron to carbon quotas from all the experiments, ranging from about 7, which is the value, if you remember from the table I showed you initially that had been published, to greater than 1,000 micromoles per mole. And the highest quotas were seen at these kind of really low bacterial biomass, and then we had lower quotas at the higher biomass. And I have some lines on here that are just pointing out with the log scale where various quotas lie, but I'm going to highlight a few others here as well. So on the bottom, this red line, this is around that 7.5 quota value that had been published prior to this study, and that was from heterotrophic bacteria in iron-limited surface waters of the Pacific. So that was kind of observed on our lower end of quotas as well. I didn't have time to show it, but there was another paper published looking at a cultured Pseudoalteromonas in 2020, and they got a quota for heterotrophic bacteria of about 82 micromoles per mole. So that's kind of mid-range in the values that we saw. And then I put some of the data I was showing you earlier that we had calculated from our uptake studies at Station Aloha. That was up here more in the like 700 to 800 range. So across all of our experiments, we kind of see values that encompass these other published values, and we saw some that were also a lot higher than this. So it seems like there's this kind of really huge range in iron quotas for bacteria. And we wanted to try to glean from this range of quotas that we were able to observe if there was some kind of minimum iron to carbon quota that seemed to be needed for cell division by bacteria. And that's what we calculated here in this red line. It was about 34 to 40 micromoles per mole. So we could interpret this to be kind of a minimum quota where like below this bacteria might be stressed for iron. And one thing that kind of supports this minimum quota value is if I put that data now on the left, and here is just our kind of iron to carbon quota now versus dissolved siderophore concentrations, kind of below this red line or minimum quota value is usually where we saw the most siderophore production in our experiments. So just to kind of like summarize everything that we saw here, what we think is happening that we observed in these experiments is that we think at these kind of low bacterial biomass range and really high iron quotas that mesopelagic bacteria are kind of storing a lot of iron. They're waiting for kind of the right moment and right conditions to get a pulse or a source of labile carbon, and then they start to grow and divide. Then when carbon substrates become available, they start kind of reallocating the stored iron into functional proteins, and that's where we see this kind of drop in quotas as they start to divide. And then when they run out of that iron, they start devoting energy to the acquisition of more iron if there's organic carbon still available. And that's where we saw this really kind of outsized siderophore production. That might be where they're approaching this kind of minimum quota that we estimated from these experiments and where they might be experiencing some iron stress. And we thought this was an interesting way to look at it, and we wanted to think about how this fits into more of an oceanographic context. And so we kind of used this minimum quota and these estimates of these iron to carbon kind of um, supply ratios that might be needed. And we kind of, from our data, predicted that heterotrophic iron deficiency should be maybe observed in the mesopelagic and the Pacific from, just below the euphotic zone to about 500 meters. And what was interesting is I think Dan Rapetta already gave a previous call and talked about this work, but his PhD students, Jay and Lydia, published this siderophore transect, this beautiful data set recently from a geotraces cruise in the Pacific. And they also saw this kind of like peak in siderophores in the mesopelagic, like we had seen originally at Station Aloha, but it was kind of a common feature throughout the Pacific. And it was most prevalent down to about 500, 600 meters. And that's kind of what we predicted based on this experimental data. So that was really neat. So what does this all mean for carbon export? I think we're not sure. It's very complicated. So for example, in our experiments, we're in the mesopelagic, inorganic nutrients are relatively high there. We're kind of in the nutrecline, and organic carbon is lower, obviously, than surface waters, but there's still some amount available. And what we saw was that when we added iron, we saw an increase in bacterial growth efficiency and increases in growth rates. So when we added more iron, there was less respiration. So does that mean there's more carbon export? Perhaps. This was interesting because there was some previous work done by Phil Tortel in surface waters, and also this work by Lauren Mank that she's done recently, more in the deep chlorophyll maximum. And she and Phil both saw actually an opposite effect. So when she added iron to the DCM, she saw lower bacterial growth efficiency. And that's what Phil saw as well. So maybe in the euphotic zone where the inorganic nutrients are low, organic carbon is available, adding iron so far has been observed to call a decrease in bacterial growth efficiency. So that's more respiration and less carbon export. So I think there's probably a lot of interesting interactions to look at and lots of future directions. So what our kind of take homes are that mesopelagic bacteria have really high iron requirements, perhaps due to some kind of storage of iron when it's available. They have this huge plasticity in their quotas. And when the iron availability is low, we saw that they dedicated a lot of energy to additional iron acquisition by producing siderophores. And so that's what we think is leading to the siderophore maxima in the mesopelagic and also these slower growth rates when they're doing that. And we don't know what the ultimate impact of this is yet on carbon export efficiency, but I do think it's something we really have to think about, especially for marine carbon dioxide removal. If we're kind of adding a bunch of iron, what is that going to do to this kind of not just the primary production, but the respiration and other side of this kind of biological carbon pump? And I think that it might vary a lot across different iron and nutrient regimes. So that's it. I'll just leave us here with some of my acknowledgements and take any questions if we have time. Thank
Thank
you.
you very much. Huge amount of stuff in there. Fascinating. Thank you. We're almost up against the half an hour, but I think we've got time for at least one quick question if anybody has one. So if people can raise their hands if they have a question that they would like to ask. There must be one out there, surely. Yeah, Alessandra.
Hi, Randy. Hope you can hear me.
Hi. Yeah.
Thanks for the great talk. Hi, everybody, as well. Nice to see these talks back, Adrian. You talked a lot about the effect of this on the carbon cycle, but what impact do you think this bacterial iron requirement is having on the distribution of iron in the ocean?
Yeah, I didn't put that in here, but basically if we use kind of our range of quotas that we saw and then like typical bacterial biomass in the mesopelagic, it could at sometimes not matter at all in terms of like the amount of the particulate iron pool, and then sometimes matter a lot. It really depends on so I think I would imagine at least thinking about like how much of the particulate iron pool this might encompass, that it would vary a lot seasonally and by region, because I think there's some times where they're in this kind of like iron storage mode and other times where they're maybe growing and dividing and things. I haven't quantified it as much in terms of the like dissolved iron uptake, just because I think we have less data for that. And so I don't know how that would translate, but I should think about that more.
Excellent. Thank you very much. We're going to have to move on, I'm afraid, but I'm sure Randy would be very happy to answer any questions that you can either feel free to put them in the chat or, contact her by email. But thank you very much for that, Andy.